As mentioned, viral structure is comprised of the nucleic acid, DNA or RNA, double-stranded or single-stranded, surrounded by a capsid, which can be arranged as helical capsomeres, icosahedral capsomeres, or complex structures. And then outside of that, some viruses are surrounded by a membrane. This membrane is what's referred to as a viral envelope and is found in only some viral particles. The viral envelope is comprised of phospholipids that are derived from the host cell membrane as the virus exits the host cell. This viral envelope can also contain viral proteins known as spikes that will help the virus attach to the next host cell it infects. The enveloped viruses that are surrounded by the membrane can protect the nucleocapsid inside. Other viruses do not have this envelope and are referred to as naked viruses. Viruses can be classified in many different ways. As we talked about, their classification usually centers around what nucleic acid type they contain inside. Based on that primary information, along with additional information about their capsid structure, number of capsomeres, and other chemical and physical properties, over 4,400 viral species have been described. Each one of these is assigned to one out of every 122 families of viruses. There is more likely to be many unidentified viral particles that are still yet to be discovered. Next, let's talk about viral replication. Viral replication has been widely studied in the T4 bacteriophage that you see here. Remember that the T4 bacteriophage can only infect E. coli cells and does not infect any other cell type. The T4 bacteriophage can infect an E. coli cell and then release or come out of the E. coli cell by causing the cell to explode or lyse. This is what's referred to as the lytic cycle of bacteriophage replication. There is also an alternative lysogenic cycle. The lysogenic cycle is when the bacteriophage enters the E. coli cell and the DNA of that bacteriophage becomes incorporated or becomes a permanent part of the E. coli cell's chromosome. Every time that E. coli cell goes to divide, it also copies the viral particle's DNA along with it unknowingly. After many rounds of cell division, you can imagine an entire E. coli population that's comprised of cells that contain a ticking time bomb of prophage bacteriophage DNA. When there is a stressor to this population and the bacteriophage realize that their host cells may die with them inside, those bacteriophage DNA molecules can be triggered to come out of the prophage state, out of the lysogenic cycle, and enter the lytic cycle, replicating the viral particles inside the E. coli cell and releasing by bursting or lysis. There are 5 main stages of viral replication that are covered in all viral replication. Bacteriophage viral replication as well as replication of human viruses. Those 5 stages are adsorption, penetration, replication, assembly, and release. All of these 5 stages are described in Table 8.3 of your text, which is a really handy study guide for your quiz. But your book also goes into more detail, showing individual pictures of these different viral replication steps. The first step is viral adsorption, otherwise known as attachment. This is adsorption, not absorption. And adsorption is when a virus attaches to the host cell surface. This is depicted in figure 8.7 for the T4 bacteriophage. Remember, T4 bacteriophage are infecting E. coli cells, and since they're infecting E. coli cells, they have a cell wall that they have to bypass as well. The T4 bacteriophage attaches its tail fibers to the cell walls of the E. coli cells. This attachment is very specific, much like a lock and key fit. If the cell walls of the E. coli cell do not contain proper receptor molecules, the T4 bacteriophage viral particles will not attach. And if they cannot attach, then they will not replicate inside that E. coli cell. The tail fiber and receptor molecule attachment, in fact, is one of the determinants of what's referred to as the host range or tropism of viral particles. The host range is defined as the cell species that the virus can infect. So for instance, rabies, which can infect humans and other mammals, has a wide host range. Having a wide or broad host range means that that particular viral particle can attach to many different cells from many different species. On the other hand, HIV has a very narrow host range because it can only infect human cells. This step of viral adsorption also dictates or determines tropisms. Tropisms are the tissue type that a viral particle can infect. So for instance, smallpox that we talked about before can infect a person through the respiratory tract, through skin-to-skin contact, and the mucous membranes of the body. On the other hand, HIV can only infect white blood cells, and even then, only specific or certain types of white blood cells. So while smallpox has a broad tropism and can infect many different tissue types, HIV has a very specific tropism and can only infect one type of cell. That's adsorption. Next comes penetration. Penetration is when the viral particle gets its nucleic acids into the cell. With T4 bacteriophage that we've been looking at, this process occurs by injection. The nucleocapsid head contains the double-stranded DNA molecule, and when the tail fibers adsorb to the cell wall, the DNA molecule inside the viral particle is injected into the E. coli cell. Human cells, which are eukaryotic, have a very different mechanism of penetration, primarily because we don't have cell walls around our cells. So there are 3 types of mechanisms whereby viral particles can penetrate into the cell. The first is endocytosis, where the viral particle essentially tricks the host cell into thinking that it's food. The host cell then engulfs the viral particle, bringing the viral particle inside the cell, eventually breaking down the capsid of the viral particle, releasing the nucleic acid into the cytoplasm. A second mechanism only works with viruses that are enveloped and already have a cell membrane. This mechanism is known as direct fusion, where the envelope of the viral particle fuses to the cell membrane of the host cell, and in doing so, releases the contents of the viral particle into the cytoplasm. Finally, there's a third type where the viral particle makes contact with the cell membrane and releases the nucleic acid contained within the viral particle directly inside the cytoplasm. The next two steps of viral replication is replication and assembly. In this case, the viral particle takes control of or hijacks the host cell mechanisms and gets the host cell to generate the different parts of the viral particle. The host cell would then make capsomeres, copies of the nucleic acids, and other components. That is what replication is. After replication is assembly, and this is when all the different parts of the viral particle are brought together to form an assembled structure, such as the nucleocapsid. This leads us to the final stage of viral replication, release. Release of T4 bacteriophage is done by lysis, where the E. coli cells are so full of viral particles or virions that the host cell erupts. This definitely kills the host cell, and the released bacteriophage viral particles can then go on to infect other host cells. This can also happen with animal viruses as well, but it only happens with animal viruses that are naked and do not need the host cell membrane. Enveloped animal viruses need to take some of the host cell membrane with them as they leave. This happens in the process known as budding. Budding is when the viral particle, the nucleocapsid, approaches the cell membrane of the host cell and causes the cell membrane to bleb outwards, eventually resulting in the nucleocapsid being surrounded by the host cell membrane itself. The host cell membrane in this case would also contain viral proteins known as spikes to allow the viral particles to then infect the next host cell. This happens with enveloped viruses like HIV, which is the picture we see over here, where HIV is budding from a T lymphocyte, and the HIV is pictured in green. This budding process, while it doesn't immediately kill the host cell, eventually results in death of the host cell because the host cell is spending all of its energy forming more viral particles instead of growing and surviving itself. All of this viral replication eventually leads to host cell damage, which is what our next topic will be in the next section of the lecture.